Hello everyone, welcome to Get My Class. So let's solve this question here. It says if the pitch of the helix is 4.8 nanometer and there are roughly 12 base pairs in each turn, calculate the distance between a base pair in helix. Okay, that is in the double helix we're talking about. Option A says 40 nanometer, option B says 0.4 nanometer, option C says 0.4 angstrom, option D says 4 nanometer. Now, what does the question say? Question says if the pitch of the helix is 4.8 nanometer and there are roughly 12 base pair in each turn, then we have to calculate the distance between a base pair, like distance between two bases in a particular strand. Now, if you see normally in the normal DNA, so this is your right-handed DNA. The general DNA what we have is right-handed helix, right? Right-handed helix. Okay, and we have, if you see the structure, this is the structure of the DNA which is present in the right-handed helix form. And if you have this pitch, this is called as the pitch. Okay, this is the pitch, and this pitch, the pitch size in normal DNA is about 3.4 nanometer. 3.4 nanometer. Okay, that is the distance between like one turn, because of starting of the turn and ending of the turn. That is what we call it as a pitch. So that is 3.4 nanometer. Okay, and the turn in one turn, there are approximately 10 base pairs. Of course, it is 10.4 or something, but then we will consider only 10 base pairs. So in one turn, there are 10 base pairs. Okay, base pairs. Okay, because we are talking about the double helix strand, so we are considering base pair. Now, if you talk about how many, like what is the distance between two bases here, or this distance you're talking about, this distance, okay? I'll change the color. So I can say that you can talk about the distance between two bases. So here, okay, here they have mentioned. It is 0.34 nanometer. How did they get? It is 3.4 by 10. Okay, so that will be, what will that be? That will be 34 by 100, and that will be your 0.34 nanometer. Okay, so the distance between two bases in the same strand will be 0.34 nanometer. Okay, so that is in the normal case. Now, what are they asking? They have given their own value. They said that the pitch is here. The pitch is what? They've given DNA, the pitch is 4.8 nanometer. And in one turn, how many? In one turn. Okay, how many are there? In one turn, there are 12 bases. Okay, or base pairs. Now, so how can you calculate if you have to speak about the distance between the two? Okay, the distance between two bases will be 4.8 divided by 12. So that will be 48 divided by 120. And that will be equal to 0.4 nanometer. Okay, 0.4 nanometer if you calculate. Okay, so let us see. Option A is 40 nanometer, which is quite large. So like 40 nanometer cannot be the distance between two base pairs. Then the nucleotides will be quite far from each other. Hence, it is ruled out. Option B says 0.4 nanometer. That is absolutely right, because our normal distance is 0.34. So here they are just increasing by two base pairs and by by another 1 nanometer, right? So it should be 0.04 nanometer. And what about option C? Option C says 0.4 angstrom. So first of all, the calculation is in terms of nanometer, hence we will totally rule it out. Option D says 4 nanometer, which is again quite more than 0.4 nanometer, hence it is ruled out. And like as per our calculation, what we got is 0.4 nanometer. That is option B. So let us conclude. What is the correct answer? Option B. I hope you understood the explanation. Thank you, everyone.